Okay, students, so this is biosafety and bioethics. The course code is BCHM315, and this is lecture 3. And the topic is classification of laboratories based on biosafety levels. This is the part 2, which includes the BSL-3 and BSL-4 labs. I am Dr. Siddiq Ibrahim, Assistant Professor, Department of Chemistry, Guwahati University of Science and Technology, and you can access the lectures at Quest Media Server Portal, which is ms.guwahati.edu.pk. Now, the topics which we will be discussing in this lecture include the barriers and procedures which are used by the laboratories to protect the workers, the lab workers, as well as others from infection. Also, we will discuss to describe the two biosafety levels, that is BSL-3 and BSL-4, and the protective measures used by each type of laboratory when handling infectious materials or biological agents. Also, we will be discussing to provide examples of the types of biological agents handled in each type of laboratory. Now, moving towards the biosafety level 3 or BSL-3 labs. Now, a BSL-3 laboratory must be used when work is done with indigenous or exotic agents that have the potential for respiratory transmission and may cause serious and potentially lethal infection. Now, the materials which are potentially infected with these agents may be studied at BSL-2 level for diagnostic purposes only, but further manipulation and experimentation then requires BSL-3 conditions. Now, agents studied in BSL-3 lab, they include Mycobacterium tuberculosis, St. Louis encephalitis virus, Francisella tularensis, Coxiella burnetii. Now, it is important to see that the image of the Francisella tularensis under the direct fluorescent antibody stain. Now, moving towards the biosafety level 3 labs. So, the primary hazard for personnel working with BSL-3 agents is the risk of infection from needle sticks, ingestion, or exposure to infectious aerosols. For example, part of public health surveillance for West Nile includes testing birds. And in August 2002, the state laboratory worker in USA cut his finger while dissecting the bird. And 4 days later, he had the symptoms of fever, myalgia, recurring sweats, and hot flashes. So, the worker and bird bird both were diagnosed with the West Nile virus. Similarly, two other lab acquired cases happened in 2002. So, this is the reason that the biosafety level 3 lab is required for dealing with such type of microorganisms. Now, in biosafety level 3, the tularemia is a common source of laboratory acquired infection. Now, what is tularemia? It is a highly infectious disease of rodents, especially the rabbits, mice, and squirrels, and sometimes it is transmitted to the humans by ticks, flies, or by handling infected animals. So the infection occurs while handling infected animals or experimenting with the cultures which may contain the tularemia, and the laboratory acquired infections were known to occur but not reportable before 9/11/2001. So after 2001, the tularemia infections were being reported, and now the tularemia is classified as a potential biological weapon. So it means that in some cases it has been observed that that microbial agent or biological agent has been transferred from one place to another place, so that the people in that place may get infected and even get killed. So that's why it is now termed as a biological weapon. Now, in case of BSL-3 labs, now moving towards the standard practices. So the standard practices which are followed in BSL-3 lab include all the BSL-2 standard practices. So all the BSL-2 lab standard practices which are followed in BSL-2 lab are also followed in the BSL-3 labs. Plus, along with the addition of strictly controlled access to the lab, which means that only the authorized personnel can enter the lab. Specific training for lab personnel in handling potentially lethal agents. So, as we know that in BSL-3 labs, we are dealing with biological agents which can cause lethal infections. So, the specific training is required for the lab personnel to obtain the training and then work in the lab. Decontaminating all waste. So when a person, a researcher is working in the lab, so after completing his or her work, the waste should be decontaminated first of all and then put in the required container. Changing contaminated protective lab clothing and decontaminating lab clothing before laundering. So it means that when you have done your work, you have completed your experiments within the lab and you are leaving the lab, so you need to change those lab clothings, as well as when you leave those lab clothings, you need to decontaminate those lab clothings before they are sent for laundering. There should be institutional policies regarding specimen collection and storage for workers to establish exposure. So whenever there is a specific sample which is coming to the BSL-3 lab and it is supposed to be a biological material. So then there should be institutional policies that how the sample will be collected, in which form the sample will be collected, and how the sample will be stored within the BSL-3 lab. Now moving towards BSL-3 level, primary barriers and secondary barriers which are used in the BSL-3 labs. So in case of the primary barriers, they are similar to the BSL-2 personal protective equipment, which is also abbreviated as PPE. So that should be used in the BSL-3 labs. And respiratory equipment, if there is a risk of infection through inhalation, so then specific respiratory equipment should be used in the BSL-3 labs when dealing with biohazardous materials. Now moving towards the secondary barriers. So again, all the BSL-2 secondary barriers are also applicable in the BSL-3 secondary barriers. The corridors of the lab should be separated from direct access to the lab. So in the coming slides we will see the layout of the BSL-3 lab, and we will be able to see that how the corridors, they are separated from direct access to the lab, and access through self-closing double doors. So there should be double doors, and they should have the ability of self-closing. Air handling system to ensure that negative air flow is maintained. So air flows into the lab. So it means that the air is not going from the lab to the outside corridors, but actually from the corridors the lab is going inside, sorry, the air is going inside the lab. So it means that there is no contamination taking place within the area or within the department as a result of negative airflow. Now the air which is pumped into the lab is not recirculated in the building. So that is also important to uh, observe, that the air pumped, so the air comes into the lab, and then uh, it would go from the exhaust system outside the building, but it cannot be recirculated inside the building. Now moving towards the biosafety level 3 laboratory layout. So this is the BSL-3 laboratory general layout, which you can see that there are certain additions in comparison to the BSL-2 lab, and all windows must be sealed which are present in the BSL-3 lab, and the autoclave should be present inside the laboratory, while in case of BSL-2 lab the autoclave was present outside the laboratory. Now access to the main area of the lab is through two sets of self-closing doors. So as here you can see that this is the first door, and then this is the entry, and then this is the second door, and then you can enter into the lab. And these doors are self-closing, as well as they have the biohazard sign posted onto them. Now the design and operational features of the laboratory must be tested and documented yearly, that the whole structure and the layout of the BSL-3 lab is intact. Seams in walls, floors, and ceilings should be sealed so that there is no provision of spread of the uh, infection within those areas. And the areas around doors and ventilation openings capable of being sealed for decontamination of the space. There is also a biological safety cabinet which is present here, and the bio-waste container is also present here, where the decontaminated bio-waste is being gathered. And within the centrifuges, they have the aerosol proof rotors so that the aerosolization of the lab should not take place with the contaminants. Now moving towards the biosafety level 4 BSL-4 labs. So in case of BSL-4 labs, the dangerous and exotic agents with high risk of life-threatening diseases or aerosol transmitted are being dealt or handled in BSL-4 labs. Also, some related agents with unknown risk of transmission are also being handled in BSL-4 labs. Now, the agents, they include all viruses along with the Marburg virus, Ebola virus, viruses that cause Congo Crimean hemorrhagic fever, and Lassa fever. So this image shows the Ebola virus from the transmission electron micrograph. Now in case of biosafety level 4 BSL-4 labs, what are the primary hazards which are possible when working in the BSL-4 labs? So there is a possibility of respiratory exposure to infectious aerosols and mucous membrane exposure to infectious droplets, as well as there is a possibility that the accidental sticks with needles or other sharp objects which are contaminated with the infectious materials may come in contact with the lab workers or researchers and infect them. So the examples which have been reported in the BSL-4 labs of BSL-4 level are in late 1960s, 25 laboratories acquired Marburg infections, including 5 deaths because they were dealing with the Marburg virus. Workers studying infected monkeys from Uganda. So the workers, they were studying infected monkeys which came from Uganda, and then ultimately some of them died. The first documented naturally occurring human case occurred in 1975, in which the human to human transfer of the virus was reported. So in case of BSL-4 labs, what are the standard practices? So the lab workers, they must receive specialized training in handling extremely dangerous infectious agents, containment of the equipment, and functions of the equipment. So it means that the lab workers who are working in BSL-4 labs, they should have the proper training of dealing with biological agents or infectious organisms, and also if there is a spill or if there is an accident, So how to control and minimize the damages of that accident or spill. Now the access to the BSL-4 lab is restricted, and the immunocompromised persons are never allowed to enter the lab. So the immunocompromised persons, they already have weak immunity, and when they enter the lab, there is a possibility that they might get infected with the organisms with which the lab workers are experimenting. Now the standard practices in BSL-4 lab include all the standard practices of BSL-3 plus some other practices. So for example, the strictly controlled access to the laboratory, so only those personnel who are authorized to work in the BSL-4 lab can work and can enter into the BSL-4 premises. Now, changing clothing before entering and exiting lab, and showering upon exiting is recommended. So it means that when you are entering the BSL-4 premises, you need to change your clothings, and after completing your work within the BSL-4 lab, when you are leaving, then again you need to change your clothings, and after changing your clothings, you are recommended to take a shower and then wear other clothings in which you can go out. And also decontaminating all material exiting facility. So you have to decontaminate all the areas of the labs with which you have touched or you have handled, so that there is no chance that some infection might spread within the labs or in the premises of the BSL-4 lab. Now moving towards the BSL-4 lab, the primary barriers which are introduced in order to avoid the direct contact between the biological hazardous agent and the personnel working in the BSL-4 lab. So for this purpose, the biosafety cabinets which are used at other biosafety levels are also used in the BSL-4 lab. A full body air supplied positive pressure personnel suit is also being worn by the lab worker when working in BSL-4 labs. Now coming towards the secondary barriers, so all physical barriers which are present in BSL-3 labs, they are also being present in BSL-4 lab. And there is an isolated zone or a separate building for the BSL-4 lab, and dedicated supply and exhaust, vacuum and decontamination systems are also available only for the BSL-4 lab. And it is recommended that the absence of windows within the BSL-4 lab, which means that there shouldn't be any windows present uh, in the structure of the BSL-4 lab. And if the windows are present, then they should be sealed and resistant to breakage. Now, as a summary, we can say that the laboratorians, they have long recognized the hazards of processing infectious agents. So whenever you are dealing with biological agents, infectious agents, so there are the dark sides of it as well, that you can also get infected with those biological agents. Now for that purpose, the biosafety guidelines have been developed to protect the workers in different types of labs which are handling the biological agents. Now, these policies are enacted through a combination of safeguards, including the engineering controls, means the shape, the layout of the building, management policies like restrict access to the labs, and work practices like you have to work in a good manner and specified according to the manuals and procedures. Now, the issue of the experimenter, it described differences between biosafety levels. So you need to understand that what is the risk associated with different types of biological agents, and because of the risk associated with those agents, the biosafety levels are also changing. So if the risk is minimum, then the biosafety level is also minimum, let's say 1. And if the risk is maximum, then the biosafety level is also maximum up to 4. Now, this lecture will help you understand that the process labs may have to undertake to identify microorganisms. So there are certain steps through which the lab personnel has to go to identify the microorganism, and for that purpose every lab cannot test for every organism, because the labs may not have the facilities, the infrastructures, and the equipment which is needed for testing or identifying that microorganisms, and for that purpose you need to go to another biosafety level lab for the identification of that microorganism. Now, moving towards the references, so these are the references for further reading and understanding, and the serial number 2, the Laboratory Biosafety Manual by WHO, which is also available online, is a very good book, very good manual, and you should um, get a copy of it, the soft copy or hard copy, and you can further understand the biosafety levels and the biosafety levels lab. Thank you.